afternoon to all the students. So, today we are going to discuss the most important topic from the chapter molecular basis of inheritance, and that topic is gene expression. Firstly, you have to know what is a gene, where is it present, how is it expressed, how many different types of genes are there based on expression, and how is it expressed at what levels, and what is the model you are going to learn to enhance the gene expression, to enhance the understanding of gene expression. What is the model you are going to learn? So, from gene expression topic, first what we are going to learn, what is a gene, what is a gene, and how many types of genes are present, how many types of genes are present, and at what levels of gene expression, and then you are going to learn on positive and negative gene expression, and then complete understanding of gene expression you will learn it with a model, that is Lac Operon model. Clear, right? This is what we are going to learn in today's topic. From cell biology also we have clearly understood what is a gene. Gene, which is addressed as a factor by Mendel, by Gregor John Mendel. In Principles of Inheritance we have learned a gene is addressed as what? Factor. Where is this gene present? If I have to consider this as a cell, consider this as a cell, and inside the nucleus of the cell, DNA is present, the genetic material, that is DNA is present, and the condensed form of DNA is condensed in the form of what? Chromosomes. DNA is condensed in the form of chromosomes. This is clear to us, no ma? Inside cell, inside nucleus, genetic material is present in nucleoplasm, and that genetic material is DNA. DNA is condensed to chromosomes. The unit of inheritance on DNA, or in simple terms, the expressed parts of the DNA, the expressed regions of the DNA or units of inheritance is known as what? A gene. Like, for example, a single DNA, multiple sites, multiple genes can be coded. One gene will be coding what? One phenotype. For example, the hair color is coded by capital B, let us suppose. And where are these present? These are two alternating forms of a single gene. One gene has two alternating forms, and these alternating forms are alleles. One gene has two alternating forms, and these two forms are alleles. Alleles can be present as in homozygous dominant alleles, heterozygous dominant alleles, homozygous recessive alleles. Likewise, alleles can be present. So, here if I have to consider two chromosomes, that is homologous chromosomes inside the nucleus itself, this is one chromosome and these are homologous chromosomes. Where do you find this homologous chromosome? At which stage of cell division? In meiosis 1, prophase 1, zygotine. Homologous chromosomes come close to each other and they form up a pair. This is called as synaptonemal complex. Why is it so? The two alternating forms of a gene, for example, capital B, which gives black color to the hair. This is the phenotypic expression, let us suppose. The two alternating forms of the gene are present at the same loci on two different chromosomes. Such chromosomes comes together at the zygotine stage of prophase 1 in meiosis 1. Okay. So, that set of chromosomes are addressed as what? Homologous chromosomes. Fine, right? And this is addressed as what? A single gene. Got it, right? In simple terms, once again let me tell you what is a gene is our first question. Where is it present actually? if I have to consider this as a cell, in a single cell, if you see this inside the nucleus and nucleoplasm, we have genetic material, and the genetic material is DNA. DNA exists in the form of chromatin. Condensed form of DNA is what? Chromosomes. The units of inheritance, that is genes, or the expressed regions on the DNA are addressed as what? Genes, which are unit of inheritance. Likewise, in a single DNA, multiple genes are present at different locations, and during meiosis 1, prophase 1 at zygotine stage, 2 chromosomes, those are homologous chromosomes, at which these alleles, what a single gene will have? A single gene will have 2 alternating forms of alleles, and they are located at 2 different chromosomes. When they are located at the same location and they are brought together at this zygotine stage, this set of chromosomes are addressed as homologous chromosomes. Got it right? And this forms up a single gene. Now, this gene have to be expressed, right, to show the predominant phenotypic characteristic. For example, we have taken the black color, right? This have to be expressed at any levels. What are the levels of gene expression we are going to learn? And for this expression, do we have any types of genes? Will all genes be permanently expressed all the time without any inducing factor? Or there are any genes which will be expressed only with the inducible factor? First, we are going to learn on what are the types of genes and what are the levels of gene expression. First, regulation of gene expression. Look into the theory also once. The mechanism by which a gene is able to express itself in the form of a phenotype in an organism is addressed as what? Gene expression. Likewise, we have 2 genes. One set of genes are constitutive genes and other set of genes are regulated genes. Constitutive genes are those genes in the body which are continuously expressed. They are not regulated by any other factors, like any other factor is not promoting the activity or inhibiting the activity. These constitutive genes which are present in the body, they are continuously expressing. Look into the theory, they are continuously expressed in all the cells of the body, also called as housekeeping genes. Constitutive genes are also addressed as housekeeping genes. Genes responsible for metabolizing glucose. This conducts what? Glycolysis. This conducts glycolysis. Genes responsible for metabolizing glucose are what? Constitutive genes only. Nothing is inducing here. Those genes are continuously expressing. Whereas second set of genes, this not always express. They do not always get expressed. Their activity is regulated. Like any other factor can induce the activity of regulatory genes, or any other factor can inhibit the activity of the regulatory genes. So, these are known as non-constitutive genes. Regulated genes are known as non-constitutive genes. Constitutive genes are known as housekeeping genes. For example, the examples of regulated genes you can say genes responsible for metabolizing lactose. The genes which are responsible for metabolizing lactose are what? Regulated genes. Clear, right? So, these are 2 different types of genes. And types of gene expression. We have learnt regulated genes. So, regulated genes can either be promoted or it can be inhibited. Based on the expression, if it is positive response or negative response, we have 2 expressions of gene, 2 regulations of gene. One is positive gene regulation, second is negative gene regulation. In positive gene regulation, remember here itself I am telling you, a single transcription unit has what? Promoter, operator, structural gene and terminator. This makes a single transcription unit. Here always to promoter, RNA polymerase will come and bind. RNA polymerase to the promoter region, RNA polymerase will come and bind, and this initiates the process of transcription. This initiates the process of transcription. And later to the operator region, if at all repressor is coming and binding, from where is this repressor coming? This repressor will come from a regulatory gene. Regulatory gene will synthesize a product or a protein that will come and bind to the operator region. Remember this all the time, either repressor or promoter that will come and bind to the operator region itself. So, here if you observe, RNA polymerase will always bind to the promoter region that will initiate the process of transcription. Repressor or activator is synthesized from the regulatory gene and that comes and binds to the operator region. If at all, though RNA polymerase binds to the promoter region, but then if repressor is binding to the operator region, this will inhibit the activity and you will not see any response at the end. There is no response only. Look into the theory also once here. The process that turns on or switches on the gene expression is known as positive gene expression. The genes remain non-expressed until or unless they are induced to do so. It is called as inducible regulation. Okay, this is known as inducible regulation. Here what happens? Either activator have to come and bind to the operator region or there should not be any repressor here. Here you see repressible gene expression. This is called as what? Repressible gene expression. This type of gene regulation will prevent the gene expression which is known as negative gene expression. Here can you see this diagrams are vice versa. Yeah, this applies to this activator is binding to the operator region and this promotes the transcription. If repressor is binding to the operator region, this will inhibit the process of transcription. So, likewise we have positive gene regulation and negative gene regulation. Positive gene regulation is known as inducible regulation, negative gene regulation is known as repressible regulation. Then here we see the gene regulation levels, at what levels the genes are regulated. Levels of gene regulation, or you can also say gene expression. Here you see genes on the DNA. Genes on the DNA, they can be expressed. This is the first level. First is transcriptional level, second processing, and third mRNA transport to the nucleus, fourth translational level. You will understand it better with the diagrammatic representation. First, genes on the DNA at the time of transcription. When primary transcript is being synthesized in eukaryotes, you can see both intron exon sequences. Intron exon sequences. These are introns intron exon. Okay. These introns are inactive sequences and this should be spliced. So, a gene can be expressed when the primary transcript is being synthesized at the time of transcription, and second at the processing level. At the processing level. What is processing level? In eukaryotes, inside the nucleus itself, we have three post-transcriptional events where the naive mRNA or the primary transcript is processed, wherein here three steps will happen inside the nucleus. First step is splicing, that is RNA splicing. Second, 5' capping. Third, 3' polyadenylation. This is the processing level. Even at this level, the gene can be expressed. And third, when the mature RNA, when the mature mRNA is formed, which is ready for its activity, that mRNA has a 5' cap and a 3' polyadenylation tail. This 5' cap is made up of methyl guanosine triphosphate, and this is completely made up of what? Exons. Okay, this is completely made up of what? Exons. Introns are completely spliced by RNA splicing process. Okay, even when this mature mRNA is formed, this RNA is ready for its activity and this will be transported from the nucleus to the cytosol. Okay. The mature RNA which has only exons, at 5 prime you have methyl guanosine triphosphate which is capping, 5 prime capping, and 3 prime polyadenylation. Such mRNA is transferred from nucleus to cytosol, okay, from nucleus to cytosol. And in cytosol at the last stage you can see protein expression. At the last stage you can see protein expression. This forms a polypeptide chain. Polypeptide chain. Got it? So, at 4 levels in the eukaryotes, you can see the gene expression or gene regulation. First, at the transcription level, when the primary transcript is formed, which is known as heteronuclear RNA, hnRNA. Second is at the processing level, which means at the time where post-transcriptional events are happening inside the nucleus, that is RNA splicing, 5 prime capping and 3 prime polyadenylation. And the third step is when the mature RNA is formed, when messenger RNA, the matured form of RNA is formed after splicing, capping and polyadenylation. Such RNA is ready for the activity and it is being synthesized, it is being released from nucleus to the cytosol. So, the third step is what? The release, the transport of RNA. And the fourth step is polypeptide chain expression, expression of a polypeptide chain or a protein expression. At these 4 levels a gene can be expressed. And then here in prokaryotes at the level of transcription, a model is being presented for the wide understanding of gene expression, that is Lac Operon model. Okay, Lac Operon model which is seen in prokaryotes at the level of transcription. Very important, multiple times asked question in the NEET or MCET exam, highly important. Try to understand. First, a single operon consists of what? Operon consists of what? Operon includes a promoter, operator, structural gene, a regulator which is near or far away from the promoter region, and lastly terminator. This forms a operon and the functions of these units are coordinated. Functions of this unit are coordinated. So, here regulatory gene, promoter, operator, structural genes, terminator, all these are units of the DNA, all these are units of the DNA of the Operon model. All the units will function in a coordinated fashion. That is the reason this is inculcated or described as Operon model. As lactose is inducing this model, lactose is inducing the model, or you can say inducing the expression of the gene. This is known as Lac Operon model. Got it right? So, a single operon includes what? Regulatory gene, promoter, operator, structural gene and terminator. All these units will perform in a coordinated fashion. Lactose is inducing this model. So, this model name is what? Lac Operon. First, to understand this better, you have to know, if I have to take a culture plate with lactic acid bacteria, the gene expression or the metabolism of this will be increased when you add lactose. We know lactose is a disaccharide. The two monosaccharides of lactose include glucose and galactose. Glucose and galactose are the two monosaccharides or monomers of the disaccharide, that is lactose. When lactose is added to lactic acid bacteria culture plate, the metabolism of LAB will be increased. How you see here? First, in the absence of lactose, how the model responds. I have said you this I gene is what? Regulatory gene. Regulatory gene. This will synthesize the repressor protein. Repressor protein. This repressor protein will always bind to which region? Operator region. Here the gene expression is switching off. This is the switch off model where repressor is binding to the operator region, the gene expression will switch off. To the promoter region, as usual, at the time of transcription, what is binding? RNA polymerase. RNA polymerase. RNA polymerase will bind to the promoter region, but then the regulatory region is synthesizing what? Repressor. Repressor binds to the operator region and this will inhibit the activity. Got it? And you have 3 structural genes here. The 3 structural genes include Z, Y and A. Z gene will be coding for beta galactosidase. Z gene will be coding for the enzyme beta galactosidase, Y for permease and A for trans-acetylase. Fine, right? So, Z beta galactosidase, Y permease and A trans-acetylase. Fine, right? So, in the absence of the inducer, that is lactose, what is happening? I gene is synthesizing the repressor, repressor binds to the operator region. Though the RNA polymerase is binding to the promoter region and initiating the activity, as repressor binded to the operator region, this will block or inhibit the process in the absence of lactose. Whereas in the presence of inducer, inducer here is what? Lactose. In the presence of lactose, what happens? The same process here, first to the promoter region, RNA polymerase will bind, which initiates the activity, and regulatory gene I will synthesize the protein, that is repressor. Repressor will bind with lactose. Repressor will bind with lactose. And next again to the promoter region, RNA polymerase will bind. RNA polymerase binds to the promoter region. As repressor is binding to the lactose, the process inhibits here itself. Repressor binds to the lactose, the process inhibits here itself. Repressor is being holded by the lactose. So, repressor will not be binding to the operator region here. Repressor does not bind to the operator. And the process gets continued, where Z gene will express beta galactosidase. This beta galactosidase will cleave the disaccharide, that is lactose, into glucose and galactose. Permease will increase the permeability of lactose into the cells, will increase the permeability of lactose into the cell. Transcription, that is and third gene is what? A, which codes for trans-acetylase. This will transfer acetyl group to beta galactosidase. What does trans-acetylase do? Trans-acetylase will transfer acetyl group to beta galactosidase. Got it? So, this is how the 3 structural genes are expressed, and this will give a polycistronic expression at the end. So, once again let us revise how the gene is being expressed in the presence of the inducer. In the presence of inducer, that is lactose, what happens? First come first in the Lac Operon model, to the promoter region RNA polymerase will bind, and regulatory region regulatory gene as usually synthesizes the repressor, but here repressor is binded to the inducer, that is lactose, wherein repressor is holded here. Next to the promoter region, again RNA polymerase is binding, and then to the operator region there is nothing to hold on this process, to block the process, because repressor is binded to lactose. So, the process continues. Z gene expresses beta galactosidase, where beta galactosidase will cleave the disaccharide that is lactose into glucose and galactose. Then Y permease will increase the permeability of lactose into the cells, and A transacetylase, it will help in transfer of acetyl group to the beta galactosidase. So, this is what you have to learn in the Lac Operon model. What type of questions you will get from the Lac Operon model? You may get firstly from this model, what is the Operon model consists of, what are the gene units that Operon model consists of, and to the promoter region what is being binded, to regulatory gene will be synthesizing what, to the operator region what is binded, what are the 3 structural genes and what are the enzymes they are being coding for, what is the activity of those enzymes, and you may also get the application based questions, what if lactose is present, what if lactose is absent. You may also get questions on constitutive genes and regulatory genes. You may also get questions on the levels of gene expression in eukaryotes. Remember that 4 levels of gene expression, first at transcription unit, second processing unit, third when the mRNA is being transported from nucleus to cytosol, fourth even at the translation point, that is synthesis of polypeptide, also gene expression you can see. This is the whole and sole concept of gene expression. In our next class, we will come up with a new topic from genetics again. Thank you.